Hello, my name is Nathan Treff. I'm going to be talking about predicting mosaicism from a Trophectoderm biopsy with next-generation sequencing technology and discuss some of the capabilities and limitations. Current use of next-generation sequencing technology by many laboratories includes using altered log2 ratios to predict mosaicism within a Trophectoderm biopsy. Mosaicism is defined as the presence of more than one chromosomal makeup within cells of an embryo. These embryos may actually possess reduced reproductive potential compared to those embryos that are considered to be uniformly euploid. This figure illustrates some of the possible ways in which an embryo might be predicted to possess mosaicism. In the first example, you can see that this particular embryo has both a monosomy of chromosome 15 and a trisomy of chromosome 15. This is a reciprocal error, and it's probably the strongest evidence for actual mosaicism to exist within the embryo. There are other examples of how mosaicism might be predicted, including this particular embryo, where in two different biopsies you can see the presence of the same abnormality in multiple cells. And another example where you observed multiple similar aneuploidies from individual cells within the same embryo. And then all the way down to what is actually used clinically, which is what we refer to as an altered log2 ratio. This is an intermediate value that may not cross a particular threshold that we would consider to be uniformly aneuploid. The main point of this slide is to illustrate the fact that among the many ways to predict mosaicism in a blastocyst, the methods currently used in clinical practice provide actually the weakest level of evidence that true mosaicism actually exists within the embryo. Despite this, there still may be ways to maximize the predictive value of methods by evaluating their performance using model systems. And there are a number of criteria that should be required before implementing these types of methodologies to predict mosaicism. And one of those is to evaluate cell line mixture models to define sensitivity and specificity. Another important experiment to consider is evaluating whether or not the original biopsy is predictive of what's remaining in the embryo. This would involve multiple biopsies after diagnosing the embryo as abnormal to begin with, and then asking whether or not those subsequent biopsies are consistent with the original prediction. Another important experiment to consider performing is to ask whether or not the prediction made in the embryo is actually predictive of the clinical outcome itself. And oftentimes we refer to these as a non-selection study, where the information that you obtain isn't used to select the embryo. And then using a way to track embryos such as DNA fingerprinting, you can then ask whether or not your prediction would have successfully predicted either a negative or a positive clinical outcome. And then finally, it's important to perform a randomized trial to demonstrate that these methodologies are actually efficacious in the way in which they'll be performed clinically. So as I mentioned, the main way in which mosaicism is predicted currently is through evaluation of what we call log2 ratios, and intermediate values might be considered mosaicism. This is an example of a result obtained using VeriSeq PGS, and you can see the copy number assignment either 1, 2, or 3 across all 24 chromosomes. And this arrow indicates a particular chromosome that doesn't actually reach the threshold for diagnosing a uniform aneuploidy, but might be considered a mosaic result. In fact, this sample was prepared in a specific way to introduce contamination. So it illustrates one of the potential ways in which you might observe something that looks like mosaicism but is actually caused by a different phenomena, in this case, contamination. Again, so these were artificially mixed samples, and you can see that most laboratories would report this result as mosaicism when in fact it was due to contamination. If it were the only embryo available, it may actually be considered for transfer. In fact, it's very difficult to actually interpret a single result as mosaicism with just log2 ratios. One advantage of other technologies such as NextCCS is the potential to use genotype information to evaluate things like contamination. You can see in the NextCCS plot below the VeriSeq plot a similar result in that you have a slightly increased copy number assignment for chromosome 18. Again, without any additional information, this might be considered a mosaic embryo. But because of this particular method's ability to perform genotyping, we can actually evaluate whether or not there is potential for contamination. In the plot below the copy number assignments, the allele ratios are shown. This is an evaluation of the presence of either one or two alleles. In a situation where you have heterozygosity, you should expect a 50-50 ratio in that one allele is present at the same quantity as the other allele. And you can see that that is the case in this example, where many of the genotype allele ratios are near this green line. In contrast, when you have a contaminated sample, it results in a skew away from a 50-50 ratio, and you start to see data points that are relatively random across this particular plot. And this actually indicates the presence of contamination, again, which is an alternative source for these altered log2 ratios. So the point here is that contamination is one potential reason why we might observe a log2 ratio that's not actually due to true mosaicism, and therefore would be a false positive diagnosis of mosaicism. Another example of how a pattern consistent with mosaicism may actually originate from something else, such as triploidy, is shown in this slide. This is an example where the copy number assignments look as though there are two copies, with the exception of one particular chromosome, chromosome 2, which has a lower relative quantity, but again not to the point where it would be considered a uniform monosomy. In this situation, it actually looks like a mosaic chromosome 2. And without any additional information other than copy number, this particular embryo would have been diagnosed as mosaic. In fact, if you look at the genotypes, they actually differ from the 50-50 ratio for the heterozygous positions in a way that's actually consistent with triploidy, where instead of having two alleles, you actually have three alleles across the entire genome, and therefore the ratio, instead of being 50-50, is 1/3 and 2/3, as shown in this plot here. Again, you can see that in many cases, the allele ratio is closer to this 33 and 66% level, and is dispersed from the 50-50 allele ratio. And this type of analysis allows you to actually identify this particular embryo as triploid, so that in fact all of the copy numbers for these chromosomes are 3, and for chromosome 2 there's actually 2 copies. Again, an example of where just simply looking at log2 ratios would have suggested a mosaic prediction, but in fact, based on genotyping allele ratios, demonstrates that this particular embryo is triploid. Although mosaicism predictions from a single Trophectoderm biopsy may actually be possible, there still are many limitations that need to be considered. We really need more research into the predictive value of these new methods for predicting mosaicism, and next-generation sequencing technologies, which also provide genotyping data, may actually help prevent false positive predictions of mosaicism. I've included 3 important references that are relevant to this particular topic. The first paper describes the use of a mixture model to evaluate the sensitivity and specificity of 2 different platforms, including the VeraSeq PGS next-generation sequencing-based methodology and quantitative real-time PCR. And one of the important components to this particular study is the ability to ask how often the technology produced a false positive prediction of mosaicism. This type of analysis is critical for evaluating predictive values. Another recent study that has gotten a lot of attention was published in the New England Journal of Medicine and suggested that healthy babies were born after the transfer of a mosaic blastocyst. And one of the issues with this particular study was that they really didn't evaluate whether or not the results that they obtained could actually have been due to false positives, when in fact the embryo was uniformly euploid to begin with, and therefore led to delivery because it was in fact euploid and not because it was truly mosaic. The third reference that I've included is just a recent review article describing some of the challenges associated with mosaicism in our field of pre-implantation genetic screening. Thank you.